The development of the cyst nematode Heterodera schachtii. In a sugar beet crop, patches of poorly growing plants may occur. The plants wilt readily in dry weather. The damage is caused by the soil-inhabiting, cyst-forming nematode Heterodera schachtii. Attacked beets have a bearded root system. White females of the nematode can be seen on the lateral roots. They are about 0.5 millimeters in diameter. The brown, dead females are called cysts. They are usually filled with several hundred eggs. Larvae in the second stage of development hatch from the eggs. They reach the soil space through openings in the cysts. The hatched larvae usually aggregate along the root hair zone of growing roots. For this film, seedlings of rape were used, which were grown in sterile nutrient agar at a temperature of 25 degrees centigrade. The infectious second-stage larva is about 0.5 millimeters long. It thrusts its mouth stylet vigorously at the cell wall. This and the following exposures are slightly accelerated by time-lapse. Now the larva has penetrated into the cell with its head. It tries to perforate the next cell wall. As soon as this happens, the turgor pressure of the cell collapses. The larva is entering through the penetration hole into the cell. Forward progression through the cell is accompanied by side-to-side movements of the head. The wall of the adjacent cell is explored by movements of the lips, which are provided with sensory organs. Soon afterwards, the nematode starts to pierce the cell wall with its mouth stylet. The arrows mark the anterior and posterior ends of the larva that has settled permanently in the root tissue. A lateral root develops near the infection site. This is shown very much accelerated. Only one photograph was taken every 6 minutes. When, after about 5 days, the larva breaks through the root cortex, it has already molted and is in the third developmental stage. The head remains inside the root. Uptake of food is rhythmical. The larva continues to grow. The posterior end of a larva, about 14 days after penetration. It now molts to the fourth developmental stage. The newly formed cuticle is now visible. After molting, ingestion of food and growth start again. During the swelling period, the larva continuously removes nutrients from the root. Food uptake stops for a short while, and then the body suddenly collapses. The larva grows, its volume increases, and the cuticle of the preceding developmental stage is burst open. Another fourth-stage larva, now just before the last molt to the female. Uptake of food stops, molting takes place. The time between root penetration and molting to a young female is about 16 days. The young female still continues to grow. The cuticle of the preceding stage bursts open. A gelatinous droplet at the hind end covers the vulva and expands as the female becomes older. The female continues to feed, but there's almost no further increase in volume. In this female, parts of the interior organs are visible. Several eggs can be recognized in the ovaries. Movements appear so quick due to high time-lapse frequency. The anterior part of a female. During ingestion of nutrients, the medium bulb, the pumping organ of the nematode, pulsates continuously. Pumping is shown here at natural speed. Nutrients are ingested exclusively from a giant cell system which is recognizable in the center of the slightly swollen root. In the vicinity of the giant cell system, several short lateral roots have been formed. A stained thin section through the giant cell system shows its internal structure. It consists of cells with partially dissolved walls. Cell walls appear thickened where they abut xylem vessels of the host plant. The thickenings are in fact wall ingrowths of the secondary cell wall. Here the short-distance transport of nutrients is enhanced. The dense cytoplasm is an indication of the high metabolic activity of the giant cells. The development to a male is also shown much accelerated. About 4 days after penetration, the larva breaks through the root tissue. A few days later, it molts to the third developmental stage. Only up to the end of this stage does the male larva ingest food from the giant cell system. Lateral roots emerge as a response to the infection. Food uptake by the male larva is also rhythmical, as shown by the typical body swellings and collapses. Molting to the fourth developmental stage is about 2 weeks after penetration. During the following 16 hours, the male larva becomes vermiform. The last moult to the male follows. For some time the male remains enclosed in the old larval cuticle. Usually the male tries to perforate the old larval cuticle for many hours. Movements are shown slightly accelerated. The head emerges through the opening. In the meantime, a male has liberated itself from the larval cuticles. The full-grown male is about 1.5 millimeters long. It moves through the agar towards a female. The female obviously emits a sexual attractant, which is perceived by the male. A male has reached the hind end of a female. The male puts out the spicules, a paired copulatory organ, and explores the genital orifice of the female. During copulation, it inserts the spicules into the orifice. Usually, a female attracts many males, and mating can happen several times. Males live only for a few days. Their only function is copulation. After mating, the male leaves the female, as shown here, much accelerated. The female, however, still feeds for some weeks and permanently produces new eggs. At the end of egg production, the female dies. Its cuticle becomes transformed into a compact brown cyst, within which the numerous eggs are well protected. The eggs inside the cyst already contain young larvae. Second-stage larvae are enclosed in these eggs. They can persist inside the eggs for many years in a dormant state. The larvae hatch when they are activated by root exudates of a host plant. These larvae will also infect host plant roots. The life cycle of Heterodera schachtii begins again.